Nowadays, colorectal cancer or CRC is the fourth and fifth most incidence and death respectively in Indonesia. Despite the slow progression of CRC, a majority of patients are diagnosed at advanced stages due to difficulties in diagnosis. Current screening methods still have several flaws. FOBT and FIT screening method using occult blood as biomarker have low sensitivity and specificity, especially at premalignant stages of CRC. Another widely used method is colonoscopy, but it comes with its own problem as it is more invasive and requires technical experts to perform the procedure. All conventional screening methods mentioned before require trained nurses or doctors to operate. So, patients need to come to a healthcare facility and spend their day there. This is troublesome to the average person, especially in rural areas, as they need to dedicate an entire day to reach healthcare facilities, undergo the procedure, and wait for the result. Therefore, an alternative solution is highly needed to enable a safer, more effective, and more inclusive way of CRC screening, especially to increase screening rates among rural populace. Thus, our project ColDBlue, colorectal cancer screening biodevices with a blue chromoprotein, was initiated. Coming with the concepts of synthetic biology, we developed a living early screening system with an engineered ECN to become the host of a Lyra biodevice that could detect miR-21 and miR-92A as the early biomarkers of CRC.
We designed and optimized engineered ECN by applying RTS on-off system production and degradation according to presence and absence of miR-21 and miR-92A. This process is done in 6 cycles, consists of testing the iGEM expression in ECN, design of early RTS, design the Lyra organ RTS system, optimize the Lyra's metrics, optimize the Lyra's regression pipeline, and lastly, evaluate the Lyra expression system.
In the first cycle, we plan to test the iGEM expression in ECN. Thus, we conduct the ECN isolation, plasmid curing of PMUT1, iGEM chromoprotein isolation, and transformation of the iGEM plasmid. Then, after we receive the transformed ECN, we try to test the plasmid stability assay, growth rate assay, and modified ECN toxicity assay. From our first cycle of experiment, we gain some knowledge that could help us in the subsequent cycles, such as the ECN could be modified by transforming iGEM chromoprotein carrying plasmid and was able to synthesize blue color chromoproteins. The second one is that iGEM chromoprotein containing plasmid is stable within ECN cells. And the third one is that there isn't any significant decrease in growth rate between control and transformed ECN cells. And lastly is that unmodified ECN is non-toxic towards CRC cell line.
In the second cycle, we assess the stability of RTS structure by examining MFA value, equilibrium concentration, secondary structure, and complex formation. In the first generation RTS analysis, we found that the equilibrium tends toward the RTS miR-21 complex, which has the most negative MFA value and an ideal secondary structure. However, there is a problem with a stop codon in the miR-21 detector region near the RBS, potentially disrupting translation. In the second generation RTS, although the MFA value is more negative and the complex tend to complement with miR-21, the structure format is not ideal. The region from RBS to the start codon is close, hindering translation, so even if miR-21 is detected, the reporter protein is not produced. But kinetic modeling suggests that the visible duration of iGEM in the second generation RTS is longer compared to the first generation. To enhance our biodevice sensitivity and resolve prior issues, we designed a Lyra or kit. This kit detects at least one of 2 CRC biomarkers, which are upregulated at different phases, in a single transcription unit to reduce cellular load. The miR-21 detector domain with a distant stop codon shouldn't disrupt translation. We initially recreate Lyra structure using a journal reference, pinpointing the RBS, start codon, and detector region. We aim for an ideal structure where the RBS to start codon in Lyra remains closed with 2 biomarkers and open with at least 1 biomarker. We then substitute detector region and modified nucleotides to enable Lyra to detect CRC biomarkers.
In the fourth cycle, we are improving Lyras by performance in its on and off states. On-level measures iBlue expression when lira and biomarkers are present, while off-level measure iBlue expression with lira but no biomarkers. To find the best sequence, we are using a data-driven approach. We transform the original definition into a simulation-based predicted secondary structure from the new pack result of the sign job. In this approach, we are specifically focusing on the RBS sequence up to the start codon. Under optimal conditions, we believe that a non-complementary structure within this region will facilitate binding to the biomarker and result in iBlue expression. Simultaneously, we aim to reduce any unwanted iBlue expression from the closed lira switch based on the hypothesis that the complementary structure within this range won't form any complex. By its predicted secondary structure, we classify parentheses as off and periods as on. We determine the on and off levels for each complex by averaging their secondary structure probabilities. This equation yields various performance metrics based on different combinations. Then we select the sequence for wet lab experiment based on one of the derived metrics, which considers complex possibilities and minimizes the on level when lira is alone. In this case, the best sequence is the second one, also known as lira-rec2. In the wet lab section, we prepared the p-mod1 as the plasmid backbone and lira-rec2 as the insert. However, we are not successful in the assembly of lira-rec2 and p-mod1. After sequencing, we found that there are multiple deletions and point mutation explaining the poor assembly of performance. We learned that a more rigorous selection scheme, better quality control, and better logistics and preparation steps is required to ensure that the recombinant plasmid is successfully transformed and reliably maintained within this chassis. After we get the best sequence, we build multivariate regression pipeline to predict on and off performance of lira switch. Using parameters of the sequence gathered, we utilize linear regression, feature selection, and AutoML to predict our approach metrics that we hope in the future should be substituted by wet lab data. We tried 3 different approaches with different metrics to improve our results, aiming for a better fit and less error. Among this approach, using feature selection saw potential in predicting weighted metrics, while the better fit came from using AutoML. However, when dealing with small dataset, we needed more complex and combined models. In conclusion, our pipeline is now ready for use with wet lab data.
To optimize the lira expression system, we employed kinetic modeling. This involves understanding the system's process, making relevant assumptions, and converting it into chemical equation. We simulated scenarios with both 2 biomarkers and a single biomarker to validate the OR gate's functionality. We use initial biomarker concentration ranging from lowest normal value to highest value of approximate short-term non-lethal limit. Here are the results. In the lira simulation, with a 10-time increase from the lowest normal value concentration using 2 biomarkers, the visible iBlue duration was approximately 14.5 hours. In the simulation, with a single biomarker at the same initial Mirna concentration, it was 14.6 hours. According to literature, food transit time from the colon star to feces is about 12 until 24 hours. The longest transit time is in the sigmoid rectum section, around 12.7 plus minus 2.1 hours, where most CRC cases occur. From this, we believe that our bioDevice can detect CRC because a 10-time Mirna concentration increase can put the iBlue duration in the 12 until 24-hour range, aligning with sigmoid rectum transit time, which is the primary site of CRC occurrence.
The Human Practice Division holds the mission and vision to connect our project with the people in need, especially the ones at risk and disadvantaged positions living in rural areas. We believe that the people who can receive the benefits of early screening using ColDBlu as a bioDevice should not be limited. In order to accomplish these goals, our project pushes forward several key values: 1. Cost-effective, 2. Easy to use, and 3. Sensitive.
We have worked following the area framework, which comprises anticipating the current situation on CRC prevalence and cases, reflecting on the improvements of previous works done, through literature reviews and conducting stakeholder interviews, engaging with stakeholders and iGEM partners worldwide through educational programs, and acting upon all the suggestions, feedback, and criticisms we have received through devising a proposed implementation. We communicated and consulted with multiple stakeholders, including public health institutions, experts, scientists or researchers, medical doctor, and family of patients.
We obtained insights on the CRC condition in Indonesia through a broader lens with the Ministry of Health of the Republic of Indonesia. We familiarized ourselves with the programs conducted in rural areas and cities to address CRC as well as the feasibility of our project implementation to the general public in a more detailed manner with the Sleman District Health Office. We acquired information on the regulations and potentials of the commercialization of our project with the Indonesian Food and Drug Monitoring Agency. We discussed the scalability of our project with PathGen diagnostic technology. We conversed about our simulation methods and analysis with the National Research and Innovation Agency. We got the knowledge about the pharmacoeconomic aspect and the formulation and manufacturing process for the proposed implementation of our project with a lecturer at the Faculty of Pharmacy and a lecturer at the Faculty of Agricultural Technology. We went over the scientific concept of our project from a clinical perspective with a medical doctor. We learned about the viewpoint and experience of a wife accompanying his late husband based in a rural area fighting CRC with a family of patients.
Education, encompassing communication and public engagement, serves as the cornerstone of our project. To achieve this, we conduct 8 activities to fulfill our objective. This program includes teaching synthetic biology to high school students, advanced science training in synthetic biology, a radio broadcasting at Suara Gama radio program to share knowledge on synthetic biology and our project. We also organize programs in rural areas, such as educating the general public in Batur village on knowing and understanding the colorectal cancer early symptom through early screening. Delivering lecture at Malang Rejo Public Elementary School with them getting to know genetic and teaching interactively at Tunas Elok Kindergarten on the basic digestive system. In addition, we host Biology National Science Olympiad 2023 laboratory research workshop to prepare high school students from all over Indonesia for Biology National Olympiad. Finally, we spread awareness and insight on social media platform Instagram, Twitter, LinkedIn with total 919 followers. One of the success indicators of our project is ability to reach various layers of the population. To achieve this, we have developed 3 inclusivity categories. First, social economic status, of which we conduct direct educational outreach to rural areas and low-income communities, and we also broadcast educational content on radio to reach areas with limited access. Second, disability, of which we have incorporated subtitles and sign language interpretation into our videos profiles, as well as design a user-friendly website to enhance accessibility. Third, origin, of which we have created 9 language options for colorectal cancer educational poster to cater to a diverse audience. The iGEM UGM team has actively collaborated with iGEM teams worldwide to strengthening relationship. iGEM UGM partnered with iGEM Patras in 2 programs. First, live tape interview, Surviving Cancer. Second, comic book on cancer entitled A Dream Fight aims to engage young audience with knowledge on cancer. iGEM UGM also teamed up with iGEM UI for webinar titled Biotechnological Advancement Toward Health. Last but not least, iGEM UGM and iGEM NYCU Taipei jointly has synthetic biology and diagnostic seminar to discuss our research in diagnostic.
To improve the development of our project, we need to consider some feature work, such as the first one is that developing a kill switch for the modified ECN. The second one is that further engaging the regulatory authority regarding the product type legislation. And the third one is that developing collaborations with the industry for the scale-up process. Acknowledging the high mortality rates of CRC, we hope that ColDBlue project can be developed and implemented as a real product to significantly improve CRC screening rates, thus lowering the mortality rates of CRC, especially in rural areas. With this project, We as a team aspire that it can prosper health and hope for us, our loved ones, and the world wider communities.